Welcome back to the deep dive. You gave us, well, a monumental stack of research on some seriously hard science, the biology of aging.
It's definitely dense stuff.
Absolutely. And we are here to give you the essential shortcut. Think of this as your guide to understanding, really, the future of human health.
And today, we're diving into a change that's so profound, it genuinely redefines what it means to be human. We're talking about the pursuit of immortality.
But not in a philosophical sense, right?
Yeah, not at all. We're talking about it as pure, solvable engineering. That's the shift.
Okay, let's kick this off with a fact that sounds completely like science fiction. I had to read this one twice from the sources.
Go for it.
The first person who will live to 1,000 years old may already be alive today.
Hmm.
And get this, they might only be a few years older than the first person to reach 150.
Right. That compression is key. It sounds wild, but it points to this acceleration we're seeing.
It's a mind-bending prediction, and it definitely demands a deep explanation. How is that even possible?
It does. And the core context, is that we're standing at this really extraordinary inflection point in history.
An inflection point. How so?
Well, for the first time, aging is being redefined. Scientifically. It's moving away from being seen as this inevitable fate.
Just something that happens.
Exactly, to being viewed as an engineering problem, something with definable, targetable mechanisms we can actually fix.
So here's the promise for you listening to this deep dive. We are going to walk you through the precise biological mechanisms. We're talking about the pills, the specific gene therapies, those protein factors.
All the tools in the emerging toolkit
that make these claims scientifically grounded. By the time we're done here, you are going to know something that, frankly, 99% of people right now consider a ridiculous fantasy.
You'll understand the blueprint.
Specifically, how scientists actually plan to outrun the calendar itself.
Our mission today, really, is to unpack this stack of advanced research. We're focused entirely on breakthrough treatments, the ones that target the fundamental causes of aging.
Not just the symptoms.
No, the root causes. Things like resetting the cellular clock, cleaning out cellular junk, even completely regenerating failing organs down the line.
I really like the analogy our sources use to frame this whole revolution. It helps make sense.
So the machine analogy.
Yeah. If your body is this complex, beautiful machine, maybe think of a high-performance engine.
Okay.
Aging is simply the accumulation of wear and tear. Molecular rust, broken parts, that kind of thing.
Makes sense. Things break down over time.
And for decades, medicine mostly tried to just slow the machine down. Better oils, maybe? Focusing on diet, lifestyle changes.
Right. Maintenance, essentially. Trying to make it last longer as is.
But the research now, it's focused on building a fully equipped pit crew.
Ah, I like that. A pit crew.
Yeah, one that can repair and actually upgrade that machine while it's still running. We're not just talking about slowing things down anymore.
No, it's about targeted interventions. Designing ways to, clean the filters, replace worn-out components, even reset the internal operating system of the cells.
The science seems clear on this, based on everything we read. Aging is a complex problem, yes, but it has engineering solutions.
That's the core belief driving the field.
But to really grasp how we get to these multi-century lifespans, these incredible predictions, we have to start with the single most audacious concept, the one that underpins this whole revolution. Okay, let's define this theoretical finish line, or maybe it's not even a finish line. Longevity escape velocity, LEV. We hear this term thrown around a lot in futurist circles. What exactly does it mean in practical terms?
LEV, it's the theoretical point where medical advances start extending your remaining life expectancy faster than time itself is passing.
Faster than time is passing.
Yeah. Imagine you age 1 calendar year. Okay. But in that same year, medical breakthroughs and therapies add back, say, 13 months of healthy life to your projected lifespan.
So you gained a month.
You literally gain ground against the clock. You're effectively aging in reverse relative to the calendar ticking by.
That idea itself is just radical. It completely transforms how we think about aging. It's not this linear decline anymore.
Exactly. It becomes potentially exponential. Instead of having this fixed, finite limit out there, the limit itself starts moving away from you faster than you can approach it.
Okay, but that's the theory. Where are we now?
Ah.
Are we making any progress towards this LEV?
We are already moving.
Yeah.
And that's a key insight people often miss. Thanks to continuous, incremental improvements, public health, better nutrition, sanitation, general medical science.
All the basics we've been doing for a century.
Right. Because of all that, for every single year that passes right now, humanity, on average, gains approximately 4 months of life expectancy.
4 months extra life expectancy for every year lived.
Correct. Which means, effectively, we only age biologically about 8 months for every 12 calendar months that go by.
Wow. Okay, so we're already running at, what, 2/3 speed compared to the clock? We're already slowing relative aging down?
Precisely. We're already on the path. And to achieve true longevity escape velocity, that rate of gain needs to hit 12 months or more per year.
So break even first. Add a month for every month that passes.
At minimum, or ideally even more, to start really pulling away from the calendar, gaining that ground. That's the escape velocity point.
And this brings us back to those, well, audacious timelines we mentioned. Dr. Aubrey de Grey, the biogerontologist, he's been the most vocal proponent of this LEV concept for years. What's his specific prediction now?
De Grey predicts there's about a 50% chance a coin flip, essentially, that humanity will reach LEV within the next 12 to 15 years.
12 to 15 years. That feels incredibly soon.
It does. And what's absolutely fascinating, and you noted this from the sources, is the rigidity of that prediction. That timeline hasn't really shifted much for him in over a decade.
Which suggests his confidence isn't based on some hoped-for breakthrough, but on existing pathways.
Exactly. His confidence stems from his SENS framework, the idea that the 7 fundamental types of aging damage are known. He believes the solutions are already conceptually mapped out.
So it's an engineering and translation problem now, not a fundamental science discovery problem.
That's his argument. He sees it as almost a certainty, just requiring the time and funding to engineer the fixes.
Meanwhile, others are even more optimistic, right? Ray Kurzweil, the futurist.
Kurzweil pushes it even further. He predicts we might reach this threshold significantly sooner, possibly within the next 5 years.
5 years. How does he justify that acceleration?
His view is rooted in the convergence of exponential technologies. He sees things like AI rapidly accelerating drug discovery and genetic engineering breakthroughs coming faster and faster. That synergy, he believes, will get us there much quicker.
Okay, so whether it's 5 years or 15, the confidence seems rooted in the fact that it's not just one magic bullet we're waiting for.
No, it's a cascade. It's multiple fundamental breakthroughs happening almost simultaneously across different areas of aging research.
And maybe the most fundamental of those breakthroughs, the one providing the first real boost to that escape velocity engine, is happening at the cellular level.
Absolutely. Reversing aging within the cell itself, that's where we see some of the most concrete progress.
Right, if we're actually going to achieve LEV, if we're going to make old bodies young again, we need a way to take an old, worn-out cell and basically hit reset.
Make it young again, functionally.
And our sources are pointing squarely at something called the Yamanaka factors as this potential master reset button. What exactly are these factors? Why are they considered so powerful?
Okay, the Yamanaka factors are 4 specific proteins. Their names are OCT4, SOX2, KLF4, and CMYC. Bit technical, but basically they're transcription factors.
Meaning they control genes.
Yes, they act like master switches, turning large sections of the cell's genome its operating instructions on or off. They were originally discovered, famously, because they could take any ordinary adult cell.
Like a skin cell or liver cell.
Exactly, a specialized cell. And they could fully reprogram it all the way back to a generic blank slate stem cell, one that could then become any other type of cell. We call those induced pluripotent stem cells, or iPSCs.
And that discovery itself was massive, right? Nobel Prize-winning stuff.
Huge, revolutionary. But, as you hinted earlier, the goal for longevity science isn't to turn all our organs back into, a mush of generic baby cells. That would be chaos.
Right, not helpful.
Absolutely not. So that's the key nuance, and this was the major breakthrough for aging research specifically. Scientists discovered they could use a partial, sort of transient application of these factors.
Partial, like not the full dose.
Or, more accurately, not for the full duration. Instead of applying them for weeks to get stem cells, they found applying them for just a few days could reverse cellular aging without wiping the cell's memory, without inducing that full pluripotency.
Okay, so the liver cell doesn't become a generic stem cell.
It becomes a younger liver cell. It retains its specific identity, its function, but its epigenetic markers, its aging clock, gets wound back.
And why is avoiding that full pluripotency, that complete reset, so critical? What's the danger?
The big danger is cancer. Specifically, inducing full pluripotency carries a significant risk of forming teratomas.
Teratomas. Those are the weird tumors with different tissue types.
Yeah, they're complex, messy tumors made up of various tissues, hair, teeth, muscle, because the cells have lost their identity and started differentiating randomly. It's a major safety concern.
So by carefully timing the exposure, just a short burst of these factors.
Exactly, just a few days. They trigger a significant epigenetic age reversal. They tune the gene networks back towards a youthful state, but they stop before the cell loses its identity and becomes dangerously unstable. It avoids that cancer risk.
And the results they've achieved with this partial reprogramming in animal models,
Yeah.
they sound genuinely astonishing. Tell us about that elderly mouse study.
It was absolutely groundbreaking. They took elderly mice, 124 weeks old. That's really quite ancient for a mouse, well past their reproductive prime.
Like 80 or 90 in human years.
Roughly in that ballpark, yes. These mice received gene therapy delivering these Yamanaka factors, usually packaged in a harmless virus, an AAV vector.
Okay. And the outcome?
Their median remaining lifespan, how much longer they lived from that point, extended by a massive 109% compared to the control group mice that didn't get the treatment.
Wait, it doubled their remaining lifespan in already old mammals?
Doubled it, yeah. It fundamentally proves the biological principle. Aging, at the cellular level at least, is not irreversible. It can be pushed back.
And critically, it wasn't just about living longer, was it? The research also showed the treatment improved their health.
That's the healthspan piece, and it's crucial. The animals weren't just living longer in a frail state. They were healthier, more active. The treatment significantly improved their frailty scores.
So better quality of life, too.
Definitely. And other labs, like Dr. David Sinclair's group, have repeatedly shown similar benefits. They've demonstrated improved function in various mouse tissues after this kind of partial reprogramming, including the optic nerve.
Actually reversing age-related vision loss in mice.
Yes, and enhancing function in the brain, kidneys, muscles. It points towards a potential full system rejuvenation, all stemming from resetting the cell's internal operating system, not just patching up one broken part.
Okay, gene therapy using these factors is clearly powerful, potentially revolutionary, but it's still complex, isn't it? It's expensive, involves viral vectors, there are hurdles there.
Absolutely. Delivery, cost, long-term safety questions.
Yeah.
Gene therapy has challenges.
So is there a parallel path? Something less invasive, maybe, than directly manipulating genes with viruses?
Yes, and this is where chemical reprogramming comes in. This is the so-called pill strategy, and it's a really vital alternative approach.
A pill to make cells younger.
That's the ultimate goal. Scientists basically reasoned, if these 4 proteins can do this, maybe we can find small chemical molecules that mimic their effects or that trigger the same downstream pathways.
And using chemicals instead of genes would bypass a lot of the issues.
Potentially, yes. Small molecules are generally easier to manufacture, deliver systemically like a regular drug, and might face fewer regulatory hurdles compared to gene therapy.
And it looks like Harvard researchers made a potentially massive step in this direction just last year, in 2023.
They did. Researchers at Harvard Medical School announced they developed chemical cocktails, mixtures of specific drugs capable of achieving similar cellular rejuvenation in human cells in lab dishes.
Without the Yamanaka genes.
Without the genes. They screened thousands of chemicals and identified 6 specific cocktails. They actually termed this the EPOC method, epigenetic programming of old cell health.
EPOCH. Okay. And what did this EPOCH method do?
It was shown to restore youthful cellular profiles looking at gene expression patterns and actually reverse aging characteristics in human cells in less than a week.
In less than a week, with just chemicals.
That's what the study reported. They're looking at chemicals that seem to reverse the transcriptomic age, the patterns of gene activity in human cells remarkably quickly, and importantly, again, without inducing that dangerous pluripotency state.
How do they work, roughly?
Yeah.
If it's not the factors themselves.
They seem to work by modulating the enzymes that control epigenetics, the chemical tags on DNA that determine which genes are active. They're essentially tuning the epigenome back to a younger state using chemicals instead of proteins.
Our sources explicitly state this approach offers, and I quote, the potential to reverse aging with a single pill.
That's the headline, and it's a huge development. It offers a potentially scalable, maybe less expensive, and perhaps safer path towards reversing cellular age. If this pans out in further studies and eventually humans, it could dramatically accelerate the timeline for reaching LEV.
Okay, so while the Yamanaka factors and EPOCH chemicals focus on resetting the healthy parts, making good cells young again, There's another massive area of research focused entirely on, well, waste management, cleaning up the systemic garbage.
That's a good way to put it.
Let's talk about the senescence problem, or as they're often called, these zombie cells. What are they?
Right, senescent cells or zombie cells. These are basically damaged cells. They've accumulated too many errors. Maybe their DNA is too damaged or their telomeres are too short.
So they stop dividing. To prevent becoming cancers, maybe.
Exactly. It's a protective mechanism initially. They enter this state of permanent cell cycle arrest. They stop dividing. But crucially, they refuse to die. They don't undergo apoptosis or programmed cell death like they should.
They just hang around, undead, hence zombies.
Precisely. And they accumulate as we age. They build up exponentially, especially in certain tissues like fat, liver, lungs, skin.
They sound harmless enough if they're just sitting there not dividing. But you mentioned earlier they're actively toxic. How does that work?
Yeah, they are far from harmless bystanders. They become highly active secretors. They start pumping out this really noxious cocktail of inflammatory molecules, growth factors, proteases, enzymes that break down tissue.
A chemical soup.
A bad one. It's collectively called the senescence-associated secretory phenotype, or SASP.
SASP.
And this SASP is the real problem. It leaks out, damages the surrounding healthy tissue, creates this chronic low-grade inflammation throughout the body.
The inflammaging we hear about.
It's a major contributor to inflammaging, yes, and it actively accelerates the aging process systemically. This SASP directly links cellular senescence to most major age-related diseases: arthritis, atherosclerosis, neurodegeneration, cancer promotion.
Wow. So these zombie cells aren't just passive markers of aging, they're active drivers of it.
Absolutely. They create a toxic neighborhood that makes everything else age faster.
Okay, so the obvious therapeutic strategy then becomes find a way to selectively eliminate these zombie cells. Get rid of the toxic factories.
That's exactly the goal of senolytic therapy. Senolytics are drugs specifically designed to target and kill only these senescent cells while leaving healthy, normally dividing cells completely untouched.
How do they do that? How do they know which cells to target?
Senescent cells have unique survival pathways switched on precisely because they're resisting apoptosis. Senolytics exploit these specific pathways, essentially pushing the senescent cells over the edge into cell death, while normal cells aren't reliant on those same pathways and aren't affected.
Clever. What are the most promising senolytics right now?
The most studied and perhaps most promising combination pairs dasatinib, which is actually an existing leukemia drug.
Oh, repurposed from cancer treatment.
Yes, with quercetin, which is a very common natural flavonoid found in many plants like onions and apples.
Dasatinib and quercetin, D plus Q.
T plus Q, exactly. That combination seems to hit multiple senescent cell types effectively.
And again, the animal results with D plus Q were really persuasive, weren't they?
They were dramatic. Mice treated with D plus Q late in life again, when they were already equivalent to maybe 70 or 80-year-old humans.
This intervening when aging is already advanced.
Right. They saw significant extensions in healthy lifespan, maybe 5 to 6 years equivalent in human terms. But more importantly, they showed major functional improvements, better mobility, improved heart function, reduced frailty, less osteoporosis, less cataract formation.
So clearing out the toxic SASP allows the remaining healthy tissue to function better again. It removes the brakes.
That's the idea. You remove the source of chronic damage and inflammation, and the body's inherent repair mechanisms can work more effectively.
And beyond these systemic whole-body treatments, we're also seeing more immediate functional applications emerge, especially around healing.
Yes, because senescence is known to actively interfere with tissue repair processes, like wound healing. It slows everything down.
Makes sense if they're spewing out tissue-degrading enzymes.
Exactly. So some recent research just this year in 2024 looked at topical application, applying a different senolytic called ABT-263 directly onto skin wounds on aging mice.
Okay, putting it right where it's needed.
And the results were striking. The topical senolytic clearly cleared out senescent cells in the wound area and dramatically accelerated the healing process.
How much faster?
The data showed that by day 24, 80% of the mice treated with the senolytic had fully healed wounds. In the untreated control group, only 56% had healed by that same time point.
That's a significant difference. Direct functional proof that removing senescent cells can restore the repair capacity of old tissue.
It really is. It's a very tangible benefit.
Okay, but again, those mouse results sound amazing, doubling lifespan, accelerating healing. We have to ground this in human reality. The human trials of senolytics so far, have been a bit more modest, haven't they? We need to understand that translational gap. Why isn't it working as dramatically in people yet?
That's a critical point. Yes, the human results have been more subtle. For example, a recent trial looked at D plus Q for bone health in older women.
Okay.
They did confirm that the drug successfully cleared senescent cells. They could measure markers going down, and they saw reductions in circulating inflammatory SASP factors. So biologically, it was working as expected.
But the clinical benefit.
The overall physical improvement, like changes in bone density or physical function, was quite limited compared to what you see in the mouse models. It was there, but subtle.
Why the difference? Why such a gap between mice and humans here?
It's likely a combination of factors. Dosing is a huge challenge, finding the right dose that kills senescent cells but doesn't cause toxicity in humans. Delivery is another, getting the drug to the right tissues effectively. And drug metabolism, humans clear these drugs much faster than mice, so maybe the exposure isn't long enough. Plus, human aging is just vastly more complex than in a lab mouse.
Right. We live longer, have more environmental exposures.
Exactly. So translating these promising preclinical findings into robust human therapies is still a major ongoing effort.
And this ties directly back to that critical regulatory challenge we keep mentioning, the FDA problem.
Yes. Aging itself is not yet recognized by the FDA or most regulatory agencies globally as a medical indication. As a disease you can treat.
So you can't get a senolytic drug approved simply to treat aging.
No. You have to target a specific, recognized age-related disease where senescence is known to play a role, like osteoarthritis or idiopathic pulmonary fibrosis or chronic kidney disease.
So researchers have to pick one specific disease endpoint, even if the drug is designed to fight the general aging process that underlies many diseases.
It forces them to be incredibly creative, and it significantly slows down development. They have to design trials around specific FDA-approved endpoints like reducing knee pain and osteoarthritis, or improving lung function and IPF, even though the true potential of the drug might be much broader, targeting the generalized process of aging itself.
That sounds like a major bottleneck.
It is. It adds years and millions of dollars to the development cycle for any true anti-aging intervention. Overcoming this regulatory hurdle is almost as important as the scientific breakthroughs themselves.
All right, moving beyond fixing individual cells with reprogramming and cleaning up the zombie cell mess with senolytics, we need to talk about broader systemic repair. Things that affect the whole body's function, like the engine's fuel efficiency, its growth regulation, and the integrity of its core programming.
Good analogy. We need to tune up the whole system.
Let's start with that metabolic tune-up, specifically targeting something called NAD plus ASH. What is NAD plus and why is it so important?
NAD+, that's nicotinamide adenine dinucleotide, is, you can think of it as a master regulator molecule for metabolism and cellular signaling. It's absolutely crucial for basically two main things.
Okay, what are they?
First, generating cellular energy. It's essential for the process in the mitochondria, our cellular powerhouses, that converts food into ATP, the cell's fuel.
So fundamental energy production.
Absolutely fundamental. Second, it's required to activate a class of proteins called sirtuins. And sirtuins are critical players in DNA repair, stress resistance, and overall cellular maintenance.
Energy and repair. Sounds pretty important.
Extremely. And the problem is, as we age, our levels of NAD+ just plummet.
They go down. How much?
Dramatically. Studies suggest levels can decline by 50% or even more between young adulthood and middle age or older.
A 50% drop in this critical molecule. What does that do to the cell?
It leads directly to mitochondrial dysfunction. The powerhouses start failing, they produce less energy and more damaging byproducts, and it leads to an inability to effectively repair accumulating DNA damage because the sirtuins aren't getting activated properly.
So the decline in NAD+ is a direct cause of key aging hallmarks.
Yes. The scientific consensus now is pretty strong on this. Our sources state quite clearly, reduced NAD+ levels cause aging and aging-associated disease. It's seen as a core driver because it compromises the fundamental energy and maintenance infrastructure of the cell.
Okay, so if losing NAD+ is bad, the obvious next step is trying to boost it back up. For years, we've heard about supplements, precursors like nicotinamide riboside, NR, or nicotinamide mononucleotide, NMN. What have those actually shown?
Those precursors do seem to help restore some aspects of energy metabolism, at least in animal models and some human studies. For instance, NR has been shown to help preserve brown fat tissue.
Brown fat, that's the good fat.
Yeah, the metabolically active fat that helps burn calories and generate heat. It declines severely with age, and NR seems to help maintain it, which is potentially beneficial for metabolic health.
But there seem to be limitations with these simple precursors, right? They don't always work as well as hoped, especially in older individuals.
That's true. Simple precursors often face a bottleneck. The body's ability to actually convert them into NAD+ and utilize them efficiently can also decline with age. It's not just about providing the raw materials.
Why is that? If you give the body the building blocks, shouldn't it just use them?
It's like an assembly line problem. Aging doesn't just deplete the raw materials, NAD+. It can also slow down or damage the machinery, the enzymes that process those precursors and build the final product.
Okay. So just throwing more raw material at a broken machine doesn't necessarily fix the output.
Exactly, which led to a newer approach described in a 2024 study. They took a more systems-based view. Instead of just giving a single precursor like NR or NMN, they developed a supplement formulation designed to target multiple points in the NAD+ production pathway simultaneously, supporting the enzymes, providing cofactors, basically optimizing the entire assembly line, not just one input.
And did that work better?
According to the study, yes. This systems-based approach resulted in significant and, importantly, more lasting increases in blood NAD+ levels in humans within just 2 to 4 weeks. It suggests a potentially more effective way to restore cellular energy capacity, especially in aging systems.
Okay, that covers boosting cellular power.
Right.
Now let's look at growth regulation, specifically this pathway called mTOR. This one always feels like walking a tightrope.
It absolutely is the ultimate biological tightrope walk. mTOR, that stands for mechanistic target of rapamycin, is a central pathway that senses nutrients and regulates critical processes like cell growth, protein synthesis, and proliferation.
So essential for development when we're young.
Definitely. We need it active to grow and build tissue. But the issue is chronic high activation of mTOR later in life seems to accelerate aging.
Why? Too much growth signal.
Sort of. It promotes cellular turnover, but maybe inefficiently. It can contribute to hyperproliferation, exhaustion of stem cells, and inefficient metabolism. It's like keeping the growth signal stuck on high when the body should be focusing more on maintenance and repair.
And rapamycin is the famous drug that targets this pathway.
Yes, rapamycin is the classic mTOR inhibitor. It's well known for dramatically extending lifespan in model organisms, flies, worms, and especially mice. It's one of the most robust lifespan extending compounds known in the lab.
Okay, it works great in mice. What about in humans? Are there proven benefits?
There are. Short-term treatment with rapamycin, or related drugs called rapalogs, has shown clear and measurable benefits in elderly adults, specifically in improving immune function.
How does it improve immunity if it's an inhibitor? Isn't rapamycin used as an immunosuppressant?
Great question. Yes, at high continuous doses, it is a powerful immunosuppressant used in transplant patients to prevent organ rejection. That's the tightrope.
So how can it improve immune function in the elderly?
It seems that short-term or intermittent low-dose rapamycin acts differently. It seems to selectively clear out old, dysfunctional, senescent-like immune cells and allow for the regeneration of fresh, naive immune cells. So it sort of rejuvenates the immune system.
Like pruning the old branches so new ones can grow.
Exactly. Studies have shown it can boost the response to vaccinations, like the flu vaccine, in elderly individuals. That's a tangible health span improvement, better protection against infection.
But that risk of immunosuppression at higher doses must be a major concern. How do researchers manage that risk, that tightrope walk, when thinking about anti-aging applications?
That is the absolute core challenge.
Right.
Chronic high-dose use is definitely associated with serious side effects, not just immunosuppression, but also metabolic disturbances like hyperglycemia, mouth ulcers, delayed wound healing.
Not ideal for a longevity drug.
Not at all. So researchers are aggressively exploring different dosing strategies to find the sweet spot.
Yeah.
The focus is on intermittent low-dose protocols,
Like taking it once a week.
or less.
Yes, exactly. The kind of protocols being explored involve things like maybe 5 to 7 milligrams taken once a week, or perhaps even 10 to 15 milligrams taken only biweekly.
The idea being to get a temporary pulse of mTOR inhibition.
Precisely. A short pulse to signal the body.
Yep.
Okay, stop the excessive growth signals for a bit. Focus on cleaning up and repair. Then you back off completely, letting the system recover before the significant side effects can accumulate. It's about getting the benefit without paying the high price of continuous inhibition.
Very clever. It's really about engineering the use of the drug.
It's pharmacological engineering trying to finesse the biology.
Okay. Metabolism, growth signals. Now let's look at the actual structural markers of aging in our core programming. The genomic clocks. Telomeres.
Right, telomeres. Yeah. These are the protective caps at the ends of our chromosomes. You can think of them like the plastic tips on shoelaces.
Aglets, exactly. They protect the important coding part of the DNA from fraying or getting damaged every time a cell divides.
And they get shorter over time.
Yes, with each cell division, a little bit of the telomere end isn't copied, so they progressively shorten. Eventually, when they get critically short, the cell receives it as a major damage signal.
And that triggers.
It triggers either senescence, becoming a zombie cell, or apoptosis, programmed cell death. So telomere length acts as a kind of cellular aging clock, tracking the replicative history of a cell line.
So the obvious question is, can we actually extend these caps? Can we rewind that specific clock in living tissues?
That's been a huge goal. And yes, there's progress. The body actually has a natural enzyme called telomerase that can rebuild telomeres, but it's mostly switched off in adult somatic cells.
To prevent cancer, presumably. Unlimited division?
That's the prevailing theory, yes. Another trade-off. But researchers found a small molecule chemical designated TERT activating compound, or TAC. TERT is the active component of the telomerase enzyme.
So this chemical switches telomerase back on.
It appears to activate it. And studies showed that giving this TAC compound to aged mice actually extended their telomeres.
Did it have functional benefits?
It did. The mice showed improved memory and coordination, better muscle strength, and interestingly, a boost in the growth of new brain cells in the hippocampus, an area crucial for memory. It suggests activating telomerase carefully could have systemic anti-aging effects.
So that's a chemical activation of the body's own built-in telomere repair system. But we've also seen reports, quite surprising ones, of a purely physical approach yielding dramatic results. Hyperbaric oxygen therapy.
Yes, that study out of Israel generated a lot of buzz. It was quite disruptive. They used a specialized protocol of hyperbaric oxygen therapy, or HBOT.
Which is basically breathing pure oxygen in a pressurized chamber, like divers use for decompression sickness.
Similar principle, yes, but a specific protocol. And they gave this to healthy adults aged 64 and older for 3 months. The results were remarkable.
What did they find?
They found that this HBOT protocol significantly increased telomere length in the participants' immune cells by over 20% on average.
A 20% extension of telomeres just from breathing oxygen in a pressurized room.
Oh.
How?
The theory is related to the pressure swings. The protocol involves alternating between high oxygen pressure inside the chamber and normal air pressure. This rapid swing between hyperoxic, high oxygen, and normoxic, normal oxygen states seems to act as a significant but controlled physiological stress.
A good stress, hormesis.
Potentially, yes. This intermittent stress signal is thought to force the cells to upregulate their protective and repair mechanisms, including, apparently, telomerase activity.
And it wasn't just telomeres, was it? Didn't it also affect senescent cells?
Yes, that was the other striking finding. The same HBOT protocol also led to a significant reduction in the population of senescent immune cells by up to 37%.
Wow. So this single, non-invasive physical treatment effectively reversed 2 major biological aging markers, telomere shortening and senescent cell accumulation simultaneously.
It was a really powerful demonstration that you might be able to trigger profound biological rejuvenation effects without necessarily needing complex drugs or gene therapies, though scaling HBOT is its own challenge. It highlights how interconnected these aging pathways are.
Okay, we've covered some really specific, exciting biological breakthroughs, reprogramming cells, clearing zombies, tuning metabolism, extending telomeres. But we need a bigger picture, like a master maintenance manual for the aging body.
A unified theory of repair.
Exactly. And that really brings us back to Dr. Aubrey de Grey and his SENS framework, Strategies for Engineered Negligible Senescence. This isn't just one approach, it's a whole philosophy, right?
It is. The SENS philosophy is fundamentally an engineering approach to aging. It explicitly rejects the idea that aging is some single, mysterious, programmed process we need to slow down. Instead, it treats aging purely as the accumulation of various types of physical damage, like a machine accumulating rust and wear. And if it's damaged, the SENS argument goes, then it must be repairable with the right technology.
So it's about identifying all the types of damage and then engineering specific fixes for each one.
Precisely. SENS provides the blueprint, the comprehensive catalog of what goes wrong. It identifies 7 primary categories of aging damage, the 7 pillars of damage, or 7 deadly SENs, as they're sometimes called.
Okay, let's quickly detail those 7 pillars, because this is essentially the comprehensive engineering checklist we need to solve to achieve true biological negligible senescence, according to this framework.
Right. Pillar number 1 is cell loss and cell atrophy. Simply put, or heart muscle cells after a heart attack, and the body fails to replace them adequately. The fix involves things like stem cell therapies to replace lost cells.
Makes sense. Number 2.
Second is nuclear DNA mutations. These are mutations in the main DNA in the cell nucleus. While most are harmless, some can lead to cancer. The SENS approach here focuses on things like boosting DNA repair, maybe whole-body gene therapy eventually, or even more speculatively, backups of the genome.
Third pillar.
Third is mitochondrial DNA mutations. Mitochondria have their own small loops of DNA separate from the nucleus. Mutations here directly compromise cellular energy production and increase damaging oxidative stress. The proposed fix is complex, maybe moving copies of mitochondrial genes into the nucleus where they're better protected.
All right, so those first 3 deal with cell numbers and the integrity of both nuclear and mitochondrial genomes, the power systems.
Correct. The fourth pillar we've already discussed extensively, senescent cells. The zombie cells accumulating and spewing, SASP. The fix here is senolytic therapies to clear them out.
Okay, number 5.
Fifth is protein crosslinks. This is where proteins in the spaces between cells chemically bond together improperly over time, often due to sugar molecules. This causes tissues to stiffen.
Like hardening of the arteries.
Oh,
Or cataracts,
yeah.
or skin losing elasticity.
Exactly. Arterial stiffness, cataracts, skin wrinkles, these are classic examples of extracellular crosslinking. The fix involves developing drugs or enzymes that can safely break these specific crosslinks.
Got it. 6th pillar.
Sixth is intracellular protein aggregates. This is junk inside the cell. Misfolded proteins, metabolic waste products that the cell's internal garbage disposal systems, like the proteasome or autophagy, fail to clear out effectively. They just build up inside.
Like cellular hoarding.
Yeah.
And the 7th pillar, which sounds related but is outside the cell.
Yes, the seventh is extracellular protein aggregates. This is misfolded junk accumulating outside cells in the spaces between them. The absolute classic example here is the amyloid beta plaques and tau tangles that build up in the brain in Alzheimer's disease.
Okay, so these last 2 pillars, 6 and 7, are really about the breakdown of the body's waste disposal and quality control systems for proteins. This protein folding crisis or proteostasis collapse.
That's arguably one of the most complex infrastructure challenges in aging. Proteostasis refers to the whole network of processes that control the folding, maintenance, and degradation of proteins in the cell. It's about maintaining protein quality control.
And this system just breaks down as we get older.
It degrades significantly. The balance tips towards accumulation of misfolded, non-functional, and often toxic proteins, both inside and outside cells. This proteostasis collapse is now recognized as a major contributor to nearly all major chronic age-related diseases, especially neurodegenerative ones.
And the research is getting really specific now, isn't it? Recognizing that this protein junk problem isn't uniform across the body.
Exactly. That 2024 study we mentioned highlighted how protein aggregation during aging is highly tissue-specific. What kind of junk accumulates in an aging kidney is very different from the junk accumulating in an aging brain or muscle.
So you need targeted approaches depending on the tissue.
Potentially, yes. And another really interesting finding from that study was that often these aggregates aren't even formed from mutated proteins like in some genetic diseases. Often they're made of normal, wild-type proteins.
Just regular proteins that start clumping up. Why?
Because the overall environment has changed. The quality control systems are failing, the cellular environment is stressed, and even normal proteins start to misfold and aggregate simply because the cleanup crew isn't working properly anymore.
So the therapeutic focus has to shift then. It's not just about targeting one specific bad protein, like amyloid beta.
Right. It also has to be about bolstering the cell's universal cleaning systems, enhancing autophagy, the process where cells eat their own internal garbage.
Cellular recycling.
Exactly. And improving the function of the proteasome, the molecular machine that chews up smaller broken proteins. The strategies involve developing targeted small molecules or maybe gene therapies that can specifically boost the activity of these waste management pathways throughout the body or in specific tissues.
Okay, so SENS gives us the map of damage types. We're developing therapies for each: cell replacement, DNA repair, senolytics, cross-link breakers, ways to boost protein cleanup. But what about the ultimate component failure? When a whole organ just gives up entirely, repairing individual cells might not be enough then.
That's where the field of regenerative medicine, specifically organ regeneration and 3D bioprinting, becomes absolutely essential for achieving truly indefinite health span. If you can replace failing organs on demand, that removes a major ceiling on lifespan.
What's the core goal of bioprinting here?
The goal is simple in concept, but revolutionary in execution. overcome the massive shortage of donor organs, and completely eliminate the risk of immune rejection by creating fully functional replacement organs using a patient's own cells.
Personalized organs built from scratch.
Exactly. You take a small sample of the patient's cells, maybe reprogram them into stem cells, expand them, mix them with a supportive bioink, and then use a specialized 3D printer to layer them down onto a scaffold, guiding them to form the complex structure of the desired organ.
It sounds like science fiction. What if we actually successfully bioprinted already? Really
hmm
making real tissues?
We've moved far beyond just theory. Real progress has been made. Researchers have successfully bioprinted and sometimes even implanted simpler structures, like multi-layered skin grafts, vascular grafts, segments of blood vessels, pieces of bone, patches of functional heart tissue, tracheal splints to support airways, and cartilage structures like ears or knee minutiae.
So simpler tissues and components are achievable now. What's the hurdle for printing a complex, solid organ like a kidney or a liver or a heart?
The biggest challenge remains vascularization. Getting a dense network of blood vessels to grow throughout the thick printed organ structure is incredibly difficult. Without blood supply bringing oxygen and nutrients deep inside, the cells in the core just die off.
The plumbing problem.
It's the ultimate plumbing problem for bioprinting, but the field is tackling it aggressively. They're progressing from printing single cell types or components to trying multi-component printing different cell types and vessel structures simultaneously.
What's the realistic timeline look like? When might we see the first fully functional, personalized replacement kidney being printed?
Predictions vary, of course, but the general trajectory outlined in our sources suggests moving towards multi-organ reconstruction capabilities becoming more feasible by the mid-2040s. It's a steady progression from patches to simpler hollow organs to eventually solid, complex, vascularized organs. We're moving towards building organs that have not just the right shape, but also the right function, maybe even enhanced bionic functions eventually.
All right, let's talk about a concept that maybe more than any other sounds like something out of a Gothic novel or maybe Dracula, but it has some genuinely exciting, if controversial, scientific backing. Blood-based rejuvenation, the young blood factor.
Yes, parabiosis. It definitely captures the imagination.
Where does this idea come from, these parabiosis studies?
It comes from a surprisingly old experimental technique called heterochronic parabiosis. Heterochronic just means different ages. Basically, researchers surgically connect the circulatory systems of an old mouse and a young mouse so they share blood.
They literally stitch their blood vessels together.
Effectively, yes. It allows factors circulating in the young mouse's blood to enter the old mouse's system, and vice versa. And the results have been consistently striking, replicated in many labs.
What happens to the old mouse?
The old mouse shows remarkable signs of rejuvenation across multiple tissues. Exposure to the young blood seems to trigger the activation of new, youthful gene expression patterns. Researchers have seen reversal of age-related decline in the old mouse's brain function, muscle strength and repair, liver function, even things like hair graying.
So sharing the young mouse's blood literally makes the old mouse biologically younger in multiple ways. That's incredible. But what is it in the young blood that's doing this? Is it adding something good or maybe diluting something bad from the old blood?
That is the million-dollar question, and there's still some debate. Early research pointed strongly towards specific pro-youthful factors present in young blood that decline with age. A key candidate that got a lot of attention was growth differentiation factor 11, or GDF11.
So the idea was identify GDF11 or similar factors, synthesize them, and inject them.
That was the initial hope. However, subsequent research has been a bit more complex. Some studies couldn't replicate the GDF11 findings robustly. And newer studies are suggesting that maybe the benefit isn't just from adding young factors, but also from diluting pro-aging factors that accumulate in old blood.
Like inflammatory molecules, maybe.
Exactly. Chronic inflammatory factors, inhibitory molecules, so it might be a combination effect. Young blood provides some beneficial signals and simultaneously dilutes the detrimental signals present in old blood through a process called plasma exchange, or dilution.
Regardless of the exact mechanism, this concept is moving into human trials, isn't it? Particularly for really difficult conditions like Alzheimer's.
That's right. There have been and are ongoing clinical trials using infusions of plasma, the liquid part of blood, collected from young, healthy donors given to patients with Alzheimer's disease.
What's the hope there?
The hope is that some rejuvenating factors present in the young plasma can cross the blood-brain barrier and help reduce neuroinflammation, clear protein aggregates, or promote neuronal health in the Alzheimer's brain.
What are the results so far? And what are the challenges?
Results have been mixed and generally modest so far, not the dramatic effects seen in mice, and the challenges are significant. First, safety transfusing plasma carries risks like immune reactions or pathogen transmission. Second, practicality relying on young donors is not a scalable solution. And third, mechanism, until we know exactly which factors or lack thereof are responsible for any benefit, is hard to optimize. The real goal is still to identify the key beneficial components, synthesize them, and deliver them as a targeted standard drug rather than using whole plasma.
Makes sense.
Yeah.
Still very early days for young blood therapy in
It's
humans.
very early and quite controversial still.
Okay, moving from blood to the absolute core of cellular aging, the powerhouses. We talked about NAD plus decline causing mitochondrial dysfunction. Is there a more direct mechanical fix? Can we actually replace failing mitochondria?
Amazingly, yes. This is the field of mitotherapy or mitochondrial replacement or transplantation. It's truly revolutionary.
So you take mitochondria out of one cell and put them into another.
Essentially, yes. As we discussed, mitochondrial dysfunction is a critical hallmark of aging. Their quality declines. They produce less energy, generate more damaging reactive oxygen species, ROS. Their DNA gets mutated. Mitotherapy involves isolating healthy, functional mitochondria, usually from young donor cells or sometimes even from the patient's own healthier tissues.
Like muscle tissue might have better mitochondria than aged skin.
Potentially, yes. You isolate these healthy mitochondria and then introduce them into the recipient cells or tissues where the existing mitochondria are damaged and failing. It's like giving the cells a complete power supply upgrade.
How do you get them inside the target cells? Just inject them nearby.
Various methods are being explored, including direct injection into tissues or even intravenous injection, where the mitochondria seem to be naturally taken up by stressed or damaged cells that need them. Mitochondria actually have mechanisms to enter cells.
That's incredible. And have they shown this works? What are the benefits seen in animal models with mitotherapy?
The benefits reported are really impressive. Studies involving intravenous injections of healthy mitochondria from young mice into aged mice have shown things like enhanced metabolic function, significantly improved cognitive performance and motor skills, reduced neuroinflammation, and even increased sports endurance and muscle function.
Just from upgating the mitochondria.
It seems so. It highlights just how central mitochondrial health is to overall systemic function. This technique is now being actively explored as a potential therapy for a wide range of conditions linked to mitochondrial failure, from heart failure and stroke recovery to neurodegenerative diseases like Parkinson's and even general aging. It suggests that simply upgrading the cellular power supply could reverse many systemic aging effects.
Okay, wow. So we've got cellular reprogramming, genetic and chemical, senolytics, NAD plus boosters, mTOR inhibitors, telomere therapies, SENS damage repair, organ regeneration, young blood factors, mitochondrial replacement. This is getting incredibly complex. It sounds like the real future isn't going to be just one of these things, is it?
Almost certainly not. The overwhelming consensus is that the future lies in combination therapies, leveraging synergistic effects.
Synergistic effects, like 1 plus 1 equals 3.
Exactly. Imagine a protocol where you first use a senolytic drug to clear out the zombie cells. Then maybe you follow up with an EPOCH chemical cocktail or transient Yamanaka factor expression to rejuvenate the remaining healthy cells. Then you use an optimized NAD plus boosting strategy to power up those rejuvenated cells. Maybe add in something to break protein cross-links.
A multi-pronged attack on aging.
Precisely. Because aging itself is multifactorial, hitting multiple hallmarks of aging simultaneously is likely to be far more effective than any single intervention alone. The goal is to create a personalized cocktail of therapies tailored to an individual's specific aging profile.
But managing that level of complexity, diagnosing the specific aging issues in an individual, designing the right combination therapy, optimizing dosages, that sounds like it requires tools far beyond what a human doctor can easily handle.
Absolutely. This is where next-generation tools become indispensable. The integration of advanced technologies is what will make this transition to true precision aging medicine feasible, smooth, and crucially personalized.
What kind of tools are we talking about?
Three main ones stand out in our sources. First, artificial intelligence, AI. AI is already revolutionizing drug discovery, identifying potential senolytics or reprogramming chemicals much faster. It's also crucial for identifying complex aging biomarkers from vast data sets, and ultimately for optimizing those personalized treatment dosages and combinations based on an individual's unique biology and response.
Okay, AI for design and optimization. What else?
Second, CRISPR and other gene editing technologies. These tools allow for incredibly precise modification of specific aging-related genes. For instance, you could potentially use CRISPR to permanently insert an extra copy of the TERT gene to maintain telomere length, or to correct specific mutations that accelerate aging in certain pathways. It offers a way to make permanent, targeted genetic upgrades.
Precision genetic engineering for longevity. And the third.
Third is nanotechnology, specifically nanodelivery systems. We're talking about tiny nanoparticles designed to carry therapeutic agents, whether it's reprogramming factors, senolytics, or mitochondria, directly to specific target cells or tissues.
Why is that important?
It dramatically improves precision. You can maximize the therapeutic effect where it's needed most, while minimizing potential side effects in healthy tissues elsewhere in the body. Think about delivering senolytics directly to arthritic joints, or reprogramming factors only to aged skin cells. Nanotech is the key to targeted delivery for many of these powerful interventions.
So it's the convergence. AI designs the therapy, CRISPR might make targeted genetic tweaks, and nanotechnology delivers the treatment precisely where it needs to go.
That's the vision of integrated precision aging medicine that's emerging. It's complex, but the tools are rapidly falling into place.
Okay, we've established a pretty compelling case, based on the science, that LEV longevity escape velocity isn't just a pipe dream. It seems plausible, maybe even within the next couple of decades, if things continue accelerating.
The scientific trajectory is certainly pointing that way.
But, and this is a huge but,
Hmm?
the biggest roadblocks now might not be purely biological anymore. They seem to be shifting towards the regulatory and societal hurdles. Let's start with that massive elephant in the room, the FDA hurdle.
Right. This is the core problem, the catch-22 of aging research regulation. As we've touched on, the FDA and most similar agencies worldwide has not yet officially recognized aging itself as a single treatable medical indication or disease.
So legally, you can't get a drug approved to treat being old.
You cannot. You have to get a drug approved to treat a specific recognized disease that is associated with aging, like osteoporosis, Alzheimer's, type 2 diabetes, heart failure, cancer. The list goes on.
Even if your drug is actually targeting the underlying biological aging process that contributes to all of those diseases.
Exactly. This fundamentally impedes, or at least significantly complicates, the clinical translation pathway for any therapy designed as a true anti-aging intervention targeting the root causes. You have to pick one downstream consequence to target for your clinical trials.
So how are scientists trying to break this deadlock? How are they trying to force the conversation and actually prove to regulators that aging itself should be considered a treatable condition?
The landmark effort here is the TAME trial. That stands for Targeting Aging with Metformin.
Metformin, that's the common diabetes drug, right?
Yes, it's a very old, safe, and widely used drug for type 2 diabetes. But decades of observational data and preclinical studies suggest metformin might have broader protective effects against multiple age-related diseases, potentially by influencing fundamental aging pathways like metabolism and inflammation.
So the TAME trial isn't just about metformin itself.
No, metformin is almost the vehicle. The real goal of TAME is to establish a new regulatory paradigm. It's designed as a proof-of-concept trial to demonstrate that aging as a single process can be targeted to prevent multiple diseases simultaneously.
How is it designed to show that?
It's enrolling thousands of older adults who don't have diabetes but are at risk for other age-related conditions. The primary endpoint isn't just preventing one disease, but rather measuring the time until the participants develop their first major chronic age-related disease, things like heart failure, stroke, cancer, dementia, or death.
So it's looking at a composite endpoint of multiple aging outcomes.
Precisely. The hypothesis is that metformin, by targeting fundamental aging mechanisms, will delay the onset of any of these major age-related conditions, pushing back the entire cluster of diseases associated with aging.
And if TAME succeeds, if metformin significantly delays that first major disease.
If TAME meets its primary endpoint, it would provide the crucial legal and scientific precedent needed to finally define aging itself as a single treatable pathology, or at least a modifiable risk factor for multiple diseases. It could fundamentally change the entire regulatory landscape for longevity medicine.
It would basically open the floodgates for getting drugs like senolytics or Yamanaka factor therapies approved specifically for an aging indication.
That's the hope. Success here would unlock potentially billions in research and development funding from major pharmaceutical companies who are currently hesitant to invest heavily in anti-aging drugs due to the unclear regulatory path. It could massively accelerate the path towards LEV.
It really feels like a critical linchpin then.
It absolutely is. But even if TAME succeeds and we clear the regulatory hurdles, we immediately run into the next set of massive challenges, the ethical and economic realities of success.
Right. Let's talk about this concept of the longevity dividend versus the inequality risk. What's the potential upside?
The potential upside, the longevity dividend, is enormous. If these interventions successfully postpone or prevent major age-related diseases like Alzheimer's, heart disease, cancer, diabetes, the savings in healthcare costs would be astronomical,
Yeah.
potentially trillions of dollars globally.
And people would live longer, healthier lives, presumably staying productive longer.
Exactly. Extending healthy, productive lifespan could lead to unprecedented economic growth and human flourishing. It's the ultimate potential return on investment for biomedical research.
But the flip side, the inequality risk.
The flip side is stark. The initial cost of these highly advanced, complex, personalized therapies, especially things like custom gene therapies, cellular reprogramming, maybe bespoke bioprinted organs, is likely to be astronomical, at least at first.
Meaning only the very wealthy could afford them.
That's the deep concern. Advanced longevity treatments might initially be accessible only to the ultra-wealthy, the top 1% or even 0.1%.
And that creates a scenario that drastically exacerbates existing social inequalities, doesn't it? A future where the rich not only have more money, but potentially centuries more healthy life than everyone else.
It's a recipe for profound social stratification, potentially creating biologically distinct classes. A massive gap between those who can afford centuries of healthy, youthful life and the vast majority of the population who cannot. That raises huge ethical questions about justice and fairness.
And that potential division leads directly to the ultimate societal pressure cooker, right? How does society itself cope?
Exactly, because dramatic lifespan extension, even if eventually accessible to more people, fundamentally alters all of our existing social structures and institutions.
How so? What breaks?
Think about retirement. Current systems are built around people working for maybe 40 years and then being supported for 15, 20 years. What happens when people can work productively for 100 or 150 years and then expect retirement support for another 50 or 100? The math completely breaks down.
Okay, retirement collapses. What else?
Career progression. How do younger generations advance if senior positions are occupied by the same healthy, experienced 150-year-olds for decades? Education models, do we need continuous retraining throughout a multi-century life? Family structures, what do multi-generational families look like when great-great-great-grandparents are common?
The entire foundation of modern society work, family, inheritance, power structures, it's all predicated on our current 80, 90-year lifespan.
Completely. Society, politically and philosophically, would have to undergo a massive adaptation to accommodate populations potentially living healthfully for centuries. And we'd have to confront incredibly difficult questions about resource allocation, political power, access to these treatments, and fundamentally what the meaning and purpose of such extended lives would be, especially if that extension isn't shared equitably. Hashtag tag outro.
Wow. We have covered an incredible amount of ground today. Just to recap for everyone listening, we started with this mind-bending idea of longevity, escape velocity, LEV.
The theoretical point where we start gaining more healthy life each year than we lose to the calendar, and the predictions that we might be surprisingly close, maybe within 1 to 2 decades.
Then we dove deep into the revolutionary power of cellular reprogramming, hitting the reset button on old cells.
Both genetically, using those Yamanaka factors in a controlled, partial way, and potentially chemically through approaches like the EPOCH method, maybe even leading to an anti-aging pill someday. Both aiming to make old cells young again without turning them into tumors.
We also looked at the crucial need for waste management, tackling those toxic zombie senescent cells with senolytic drugs.
And the systemic repairs boosting NAD plus for energy and repair, carefully modulating mTOR for immune health and maintenance, and even finding ways to rewind the telomere clock using chemicals or physical therapies like HBOT.
And we wrapped up the science section looking at the comprehensive SNS framework, the engineering approach to fixing all 7 types of aging damage, and the incredible potential of regenerative medicine, like bioprinting replacement organs.
Putting it all together, the convergence of all these different technologies really does strongly suggest that aging isn't some immutable law of nature we just have to accept.
It's looking more and more like a highly complex, but ultimately treatable engineering condition.
Exactly. Our sources are becoming increasingly clear on this point. The mythical fountain of youth. It was never going to be found in some magical spring. It was always going to be built piece by piece in laboratories by brilliant scientists wielding the tools of genomics, synthetic biology, and AI.
Okay, now, we've spent this entire deep dive talking about how quickly the science is moving, how we might actually achieve something like routine age reversal, maybe even by the mid-2060s or sooner, based on these trajectories. We can, it seems, potentially fix the biology.
The path is becoming visible scientifically.
But here is the critical, provocative thought we want to leave you with, something for you to really mull over as you process all this.
If biological aging does become largely optional, if we achieve LEV within many of our lifetimes, the biggest challenge remaining might not be the science anymore. It might be the human element.
What does a 200-year healthy life actually look like in practice, in a world where maybe only 10% or even 1% of the population can initially afford the treatments that make it possible?
If we succeed in fixing the human body, but in the process we break the social contract, if we deepen inequality to an unimaginable degree, what is the ultimate value of that extended life? Who benefits and at what cost to society?
That is precisely where we are heading next time on the deep dive. We're shifting our focus from the lab bench and the biological mechanisms.
To the real-world, messy human implications.
If aging truly becomes optional, we absolutely must confront the massive socioeconomic ripple effects. We're talking the potential collapse of retirement systems, the freezing of social mobility and innovation, the extreme concentration of power and resources that could follow.
Join us next time as we dive headfirst into the potential dark side of longevity. The political turmoil, the financial instability, the profound ethical crises we risk creating if we achieve these biological miracles but fail to figure out how to share them and their consequences equitably.
You really don't want to miss this crucial follow-up as we examine the potential human costs of finally outrunning time.